lots of people and animals living in very close proximity, so we do have runoff, leaching of septic systems and things like that. So it's the most common contaminant, and it has, of course, public health, economic, and ecological impacts. The traditional method that we use, and we do monitor all the beaches up and down the coast from Minard Park up here down to Hoffmaster, and then we monitor a lot of the inland lakes like Blue Lake, Fox Lake, etc. And we always use what's called, see the marker there, the E. coli test. And whenever you're incubating and growing bacteria, there's always a lag time between when you collect your sample and you report your results. And the standard E. coli test takes 18 hours. So I collect a sample from a beach, and then I tell everybody 18 hours later that the beach was unsafe to swim in. That's not the best scenario. And the other problem with this is that E. coli is a facultative anaerobe, and what that means is it can also survive in the air. It prefers to be in your stomach without air, but it can survive in the air. So there's recently been some work that's demonstrated E. coli can survive and reproduce in sediment and soil. There's very little air in sediment. So it's really not the best indicator, and it doesn't discriminate between sources. With our DNA-based processes, we can actually look at a special microbe closely associated with a host. So everybody's intestinal tract is based on its diet and its ecology. So there's physiological differences in the gut bacteria in all these different animals. And microbial source tracking, or MST, gives us the ability to differentiate between all these cast of characters. So whenever I decide to, or we decide to look at a contamination site, we can't do a lineup like that. What we have to do is we have to test for different markers in addition to E. coli. And the markers that we typically use in our toolbox, I have a lot of choices for icons there, but we use a general ruminant marker that gives us cows and deer and sheep, just basic ruminant animals. We use a pig marker, we use a dog marker, the human marker. We have a cow-specific marker, and then we also use a gull-specific marker. And we even have a marker for all animals, all back. We're just starting to work with that one and try and use that to differentiate where we have populations of E. coli that might be growing in the environment. If we can't pin it on an animal, we may be able to see it with the all-back marker. So we're kind of looking at different combinations of markers, but these are the ones that we have available in our lab in Muskegon. And I just wanted to talk a little bit about Flower Creek, and unfortunately the screen's a little bit too small to see that, but for Little Flower Creek, we've been studying the bacteria problem there for quite a while, and the beach has, I wouldn't say maybe every year, but probably every 3 or 4 years there's beach closures. And it's usually due to Little Flower Creek. And we've actually been sampling there's a little dunal wetland, and again, there you see it. Okay, it's got an outlet, and then it's got this little wetland pocket, and that is typically high for E. coli. That's, our standard is 300, and that's running about 300 to 400 usually. And then there's a couple campgrounds up here. And we decided we would put the markers to test and look at this site in some more detail. And first of all, we sampled this area here, the wetland, and of course we found bird markers, we found the gull marker, we found human marker, we found ruminant marker. So we went upstream a little bit, and right below the parking, there's a big parking lot here, and we sampled right by the bridge. And lo and behold, the bird markers were higher there than they were in the interdunal wetland. So something's going on in the parking lot. And when you go to the parking lot, there's a dumpster, there's food all over the place, people feed the gulls. So one of our recommendations is going to be to do a little bit better job managing the trash, and that could lower the bacteria level that we're seeing in the interdunal wetland. We also found HF183, which is the human marker, and we're going to be sampling next year up into the watershed and see if there's any kind of leaking systems. I don't think there's a septic system for either one of these parks, but you know, there's piping there, there's valves to store the waste, and we want to get an idea, are there homes further up the watershed, or are these two parks, trailer camp areas and whatnot, are they sources of E. coli? And we can do that with the HF183, the human marker. And we didn't find any cow marker. The cow marker can be very specific based on the diet of the cow. If you have a CAFO that's using a high antibiotic feed, we may not be able to pick it up with the cow marker. So in order to do this, we have to get some cow poop and test it and verify that our technique works. So that's something that we're going to be doing next year too. But anyway, we're going to be doing a more expanded study of Little Flower Creek. We've been working at Flower Creek to the north, and we're also working on Little Black Creek, and that goes through Hoffmaster Park. All those little creeks have high E. coli numbers, and we're not sure if it's wildlife or it's humans. And to end, I do want to say that just because it's wildlife and humans and birds, we still need to be concerned. Everybody's heard about salmonella poisoning, right? Salmonella does not bother birds at all, but it certainly bothers us. So if you have the bird marker, you could have salmonella. Cows can carry Cryptosporidium, so you know, you probably don't have cholera, which certainly is bad, but you could have other parasites. So the question is, with these methods, they are more expensive. They cost about $100 to $500 a test, and wet weather is typically good for runoff. Dry weather is good for finding septic systems. So if we sample during wet weather, we're probably going to pick up runoff, and then if we go to sample at baseline levels, we'll probably find more HF183, so we have to do both kinds of studies. And then we also need to pair them with GIS and field surveys to help pick better locations. And I just wanted to show, this is another map from the Bass River watershed. This is in Ottawa County, and you can see that soil types have a great influence on the types of bacteria that we find. The red areas have very high bacteria and very poor soils. So septic systems in these areas are probably contributing to the overall problem. So that's it. I'll take questions later.
Good. So my name is Aaron Parker. I am an aquatic biologist with the Michigan Department of Environment, Great Lakes and Energy, or EGLE. My primary responsibility is keeping track of all of the harmful algal blooms or cyanobacteria blooms occurring throughout the state. One thing I wanted to mention though is I do have close roots to this area. My father used to live in Montague on Whitbeck Road. I also have a lot of family here too. The name Parker or Schultz is anywhere in your family tree. There's a good chance that we're related. But without further ado, there we go. Okay, so first thing I wanted to mention is the term harmful algal blooms or blue-green algae are a bit of misnomers because when you hear about harmful algal blooms or blue-green algae, really what you're hearing about is cyanobacteria. Cyanobacteria, as the name implies, are not algae, but they are bacteria. However, they're bacteria that are capable of photosynthesizing. They also compete with algae for sunlight and nutrients, and so because of that, they're considered to be part of the algal community. But it's important to remember, though, that they aren't truly algae. So not all algae that you see are the bad kind. The cyanobacteria, they've been around for a long time. They're found in very diverse environments. You can find them in soil, you can find them in marine, saltwater, People have actually done swabs on office walls and found them in buildings, but mostly where you will find cyanobacteria is in freshwater, which of course we have plenty of in this area. Cyanobacteria, like I said, they've been around for a long time. They're very good at what they do. They are capable of regulating their buoyancy up and down in a water column in order to obtain ideal amounts of nutrients and sunlight. Some of them are capable of fixing their own nitrogen out of the atmosphere if they run low in the water. And of course, large or dense blooms such as this one can be aesthetically displeasing. When these things begin to senesce or decay, they can emit strong odors. And of course, the reason why you've been hearing so much about them lately is because some of them are capable of producing toxins and other irritants. So, in general, where you will see cyanobacteria blooms like this are in water bodies that have high nutrient concentrations, when surface conditions are calm in a water body, and when water bodies are warm. So you can have a high nutrient water body, but if it's say 35 degrees and there's wind whipping across at 40 miles per hour and it's dreary out, probably not going to get one of these blooms. Now, in that same water body, if it's this time, August, and it's 95 degrees, sunny, calm, no wind, that's when you can get one of these blooms occurring. However, in the last few decades, with the invasion of zebra and quagga mussels, they've basically turned that top bullet point on its head. Because of that, because of these dry-scented mussels, we're now starting to see cyanobacteria blooms even in low nutrient water bodies. So basically, what the dry-scented mussels will do is they will ingest cyanobacteria, but then they will reject it, and the cyanobacteria can survive that process. However, they will readily feed on diatoms and green algae, so then you may end up with this disproportionate population of cyanobacteria. There's also evidence too that what the zebra and quagga mussels will do is they will excrete very nutrient-rich pseudofeces on the lake bottoms. If you recall, I mentioned earlier that cyanobacteria can regulate their buoyancy up and down in a water column. So that means the cyanobacteria can go down into the bottom, extract nutrients from those pseudofeces, and then go back up for their sunlight. Whereas other algae that may be attached to a rock right along the shoreline, they're not exactly going to be able to utilize those nutrients in those pseudofeces. This right